Muito obrigado. Thank you so much for this invitation, Nancy Guilherme. It's an absolute honor for me to present in this symposium. When it comes to morphological characters in deep animal phylogenetics, it's widely understood that molecular data and the availability of molecular phylogenies provides a considerable advantage when we reevaluate morphological characters. This is because when you have a large evidentiary body from an independent data source, you can focus exclusively on sources of discordance in morphological data. This has enabled us to revolutionize our understanding of arthropod phylogeny, both where arthropods go in the tree of life, as well as relationships within the arthropods. With the benefit of the retrospective and from the lens of molecular phylogenies, we're able to recognize that a series of characters that were previously used to unite what are now considered obsolete groupings are in fact not putative homologies. These putative synapomorphies are in fact the results of convergence. This process of reevaluating morphological characters in light of independent data sources is formally called reciprocal illumination. Put another way, hindsight is 2020. This metaphor is a little difficult to translate. The closest analogy that I have found in Spanish is something like this: Después de la guerra, todos somos generales. Everything makes sense in retrospect. These considerations weigh heavily upon the basal phylogeny of the chelicerates. Shown here is a selection of studies based on morphological phylogenies. These were largely produced by paleontologists in the last couple of years. As you will see, there's an enormous discordance across morphological phylogenies from these data sets. One study from a couple of years ago failed to recover the monophyly of the tetrapulmonates, shown in green. Another work, which is the one that most densely samples living and extinct arachnids, failed to recover the monophyly of Chelicerata, the non-sea spider chelicerates. And a work published just last year was unable to recover the monophyly of arachnids. This group constrained the monophyly of arachnids a priori in order to recover support for this preconceived relationship. Now, phylogenomics isn't that much better when it comes to discordance, with the exception of three key nodes. First, molecular data generally tend to agree that sea spiders are the sister group of a monophyletic Chelicerata. Second, phylogenomics generally does tend to recover Tetrapulmonates as a monophyletic group, like most morphological phylogenies. And third, most interestingly, molecular data has recovered scorpions as the sister group of the Tetrapulmonates. We refer to this clade as the Arachnopulmonata. It's the clade of arachnid orders that have book lungs. Now, even though morphological data have never recovered Arachnopulmonata as monophyletic, the community consensus is that Arachnopulmonates are indeed monophyletic, and this hypothesis is starting to gain traction. Now, why is that? Part of the answer has to do with the advent and the influence of new phylogenetic data classes, specifically genomics and evodevo. In a work published 3 years ago, upon the publication of the genomes of Parasteatota tepidariorum and Centroides sculpturatus, the I5K initiative was able to show that the common ancestor of spiders and scorpions shares a duplication event, that is, a whole genome duplication, to the exclusion of apulmonate arachnid groups like mites, ticks, and harvestmen, shown in the schematic by this photograph of Ixodes scapularis. Horseshoe crabs are now understood to have their own twofold whole genome duplication, at least, but this is a completely independent event and it has nothing to do with the arachnopulmonate duplication. What is the evidence for this shared duplication event in the common ancestor of the arachnopulmonates? This comes from essentially 4 different types of data sources. The first of these is the consideration of Hox clusters. Ancestrally, arthropods or panarthropods have 10 Hox genes. These occur as a single cluster in panarthropod genomes, but in arachnopulmonates, there are 2 Hox clusters, and this kind of event is usually associated with a whole genome duplication. A few years ago, I was able to show, using transcriptomes and embryonic expression data from embryonic scorpions, that scorpions have 19 Hox genes, 2 copies of every Hox gene except for Hox3, and as shown by these schematics in some of these images, these paralogs have different spatial-temporal dynamics. That is, each retained paralog has a unique location and timing of expression onset. The significance of this is that spiders do exactly the same thing. In the publication of the I5K group, we were able to show that Parasteatota also has 2 Hox clusters. It has 18 Hox genes, 2 copies of every Hox gene except for Hox3 and Fushitarazu. And just like scorpions, these paralogs have unique spatiotemporal expression patterns. The second line of evidence for this whole genome duplication comes from a broader survey of gene families and paralogy across gene families. In a work that I produced in collaboration with Alistair McGregor's lab a couple of years ago, we proceeded as follows. We surveyed all of the homeobox gene families using this particular selection of taxa. Arachnopulmonates, represented by 2 spiders and 2 scorpion genomes. We have 2 pseudoscorpions, a harvestman, Akari, represented by the Ixodes genome, and then 3 mandibulate outgroup genomes. The exercise that we undertook was to map paralogs. What you would expect if Arachnopulmonates shared a whole genome duplication is something like this. For a particular Hox gene, we might expect 2 copies for this particular gene, whereas all of the outgroup taxa have only 1. As we start to zoom out across the homeobox gene family, you will observe that the Arachnopulmonates tend to retain lots of duplicated copies of homeobox genes, whereas outgroup taxa like harvestmen or mites, or ticks will have a single copy. Pseudoscorpions, as you will notice, are very patchy in this data set, and this has to do with the use of adult tissues to represent Pseudoscorpiones. Keep zooming out further and further, and you'll notice across the entire homeobox class, this pattern is systemic. There are lots of retained duplicates of Arachnopulmonata. A third line of evidence for that whole genome duplication comes from the survey of microRNA families. These are important regulatory elements that essentially act like rare genomic changes. In a survey of microRNA families from Daniel Leite, formerly a student from Alistair McGregor's lab, this group was able to show that Arachnopulmonates, represented here by 1 scorpion and 3 spider genomes, exhibit a signature of microRNA enrichment that is consistent with the pattern of whole genome duplication. Once again, here's horseshoe crabs, and they're down here doing their own thing with their own independent duplication. The fourth and final line of evidence for that whole genome duplication comes from a survey of gene expression patterns where we know that Arachnopulmonates have a lineage-specific duplication. In a work published earlier this year by my former undergraduate, Eric Nolan, we were able to show that paralogs of leg patterning genes exhibit Arachnopulmonate-specific expression patterns. As an example, the gene shown here is doxin-1, and the dark shading is showing you where it's expressed in these scorpion embryos. Importantly, doxin-1 is expressed as a single medial band or an unbroken band in the middle of the appendage. Its paralog, doxin-2, is distinguished from doxin-1 both by the pattern of expression in the ventral ectoderm, but more importantly in the leg. It's expressed in a proximal region and as a distal band, a stronger distal band at the end of the patella. Why is this significant? It's significant because it has previously been shown in multiple spider species that the exact same pattern occurs across spiders, with 1 copy exhibiting a medial band, an unbroken band, and the other copy exhibiting the split band. By comparison to outgroup taxa, like apalmonid arachnids or the rest of the panarthropods, one of the 2 copies has what you would consider as a derived expression pattern. What's more, Eric was able to show that this pattern is exactly reflected in the gene trees of such genes when they're well sampled. So the medial unbroken band that you saw for doxin-1, all of these copies cluster together in the gene tree. There's an Arachnopulmonate specific cluster in which we have scorpions, a Europygy, and spiders. The broken copy, the one that has the split band, clusters as well as doxin-2. Here, once again, are horseshoe crabs with their own independent duplications, doing their own thing, nested inside the chelicerates. And at the base of the tree, we have apalmonid arachnid, which have single copy homologs for this gene, like harvestman and Akari. It's not just doxin that does this. Eric was able to show 3 other pairs of genes exhibit the same pattern. We won't go through lots of expression data, but take it from me, and you can examine the expression data for this. It's always the case that 1 copy of the Arachnopulmonates has this new derived expression pattern that we don't see either in the other copy or in the single copy homolog of outgroup taxa. So taken together, what we have then is this complex meta character. This whole genome duplication essentially constitutes this multi-layered synapomorphy that supports the monophyly of the Arachnopulmonates. And I won't show it here to give away the story, but these data sets have been independently validated now in whip spiders through new data sets that Guilherme Gainet, my student, is producing for Amblopygi. That's cool, but what does that really tell us about the rest of arachnid phylogeny? The purpose of today's discussion is the placement of pseudoscorpions. Pseudoscorpions are one of 3 long-branched taxa in chelicerate phylogeny, along with the mites and the ticks. They exhibit faster rates of evolution, and as a result, they exhibit an artifact called long branch attraction, where they're often drawn towards the base of the tree or with other fast-evolving taxa. So across phylogenomic studies, you will notice that pseudoscorpions generally tend to bounce around in one of 3 places: nested within or sister group to the Acari, at the base of the Arachnopulmonates, or inside of Arachnopulmonates as a sister group of scorpions. To understand what drives this instability and what contributes to this phenomenon, my student Andrew Antonow pursued this investigation by constructing first a phylogenomic data set. He generated a data set of 117 taxa representing all chelicerate orders except for Palpegrati, and he pursued specifically one effect: the effect of taxonomic sampling. It is thought that taxonomic undersampling drives long branch attraction, and we can solve this problem by breaking branches. But how effective is that solution? To check, Andrew proceeded as follows. He leveraged a data set of pseudoscorpion diversity recently generated by our co-author, Ligia Benavides, in Gonzalo Gerbet's lab. And shown here is just the tree of pseudoscorpions. Andrew simulated the effect of taxonomic undersampling by sequentially pruning branches from the base of the tree. That is, he sequentially pruned all of the superfamilies, starting from the base of the tree, proceeding distally to the chelipheroids. Here's how we'll show his data set. This box over here, T-6, represents a phylogenomic data set where everything else is represented, the rest of the chelicerate orders, but the pseudoscorpions represented only by the chelipheroids. As we move from left to right, we're sequentially going to add back in all of these basal branches until we get to T-0, the tree where all the pseudoscorpion groups are represented. The icons and colors in the boxes are telling you where pseudoscorpions went in those trees. So when you sample only, for example, chelipheroids, pseudoscorpions are recovered as the sister group of the mites. But as you sequentially add more and more basally branching taxa, pseudoscorpions pop out of Acari, and they're now sister group to the scorpions. This effect is not contingent on the completeness of the matrix. If we make a smaller matrix that is if we remove missing data by essentially picking only the genes that are very high in taxon occupancy, this effect is not mitigated in any way. That is, the only consistent solution to recovering pseudoscorpions plus scorpions appears to be if we broadly sample across pseudoscorpion diversity. Sample lineages at the base of the tree, pseudoscorpions will reliably go with the scorpions. This is a visual depiction of that result. Prune out all the branches at the base of the pseudoscorpions, and now here they are, sister group of the parasitiforms. But add in all those basally branching superfamilies, well, now here they are, sister group to the scorpions. Both of these topologies are maximally supported. They have 100% nodal support. Which of these 2 topologies is accurate? You might be inclined to say the topology on the right. Obviously, it's got more taxa. But how do we know? To answer this question, Andrew leveraged this information about the Arachnopulmonate synapomorphy. We believe that there's a whole genome duplication that occurred at the base of the Arachnopulmonates. So if pseudoscorpions are Arachnopulmonates, they should exhibit the signature of whole genome duplication. To investigate this, Andrew went to Western Australia 3 years ago and collected pseudoscorpions under the tutelage of Mark Harvey. Through that fieldwork, he was able to establish a live colony of this cernetid, Kernickokernes crassus. He obtained embryos from Kernickokernes, and he generated the first developmental data sets for pseudoscorpions. In addition, we teamed up with David and Jean Zay, who have been working on a draft genome of another cernetid, Cordylokernes scorpioides. And Andrew examined these 2 data sets, the developmental transcriptome and the genome, to ask, do pseudoscorpions exhibit evidence for that shared genome duplication? Complementing this investigation were data sets from neglected chelicerate groups, which my lab has been pushing to establish for comparative biology. From a recent phylogenomic study published this year, Jesus Ballesteros contributed the first 5 sea spider developmental transcriptomes. One of my graduate students, Emily Seddon, contributed a high-quality transcriptome for the Mygalomorph of Phylogeny hansi, shown here in scale with the embryo of Parasteatoda tepidoriorum. And another of my proteges, Guilherme Gainet, whom I'm very excited to be telling us about this topic today, generated the first 3 developmental transcriptomes for amblypygy, for whip spiders. Okay, let's get going. Do pseudoscorpions have a whole genome duplication? The first line of evidence we examined were the Hox gene complements. This is a complicated figure. Let's simplify this a little bit. See all these colored boxes? Every color corresponds to one of the 10 Hox genes that occur ancestrally in groups like velvet worms, the onychophorans, as well as the other arthropods. Horseshoe crabs are over here, and they have the signature of the twofold whole genome duplication. So they have up to between 3 and 5 Hox gene copies. Pycnogonids, incidentally, do not exhibit whole genome duplication. They have single copy genes for all of the Hox genes. That is, we do not believe that the whole genome duplication is ancestral to chelicerates. This is a derived event. Let's zoom in on just the arachnids. Kindly observe that in pseudoscorpions, we obtain duplicates of many of the Hox genes in common with the remaining arachnopulmonates. In the transcriptome of Conychokernes, we found 9 homologs of Hox genes, 6 of which are duplicated. In the genome of Cryptolechernes, all 9 of those Hox gene homologs were present as duplicates. This is to the exclusion of groups like mites, ticks, and harvestmen, which either have the ancestral 10 gene Hox cluster, or they exhibit duplications only by a tandem duplicates. That is, these groups have a single Hox cluster. Our second line of evidence comes from, again, this investigation of paralogy more broadly. So once again, we extended upon that previous analysis we generated a couple of years ago for homeobox genes. This time, though, we're going to add in pycnogonids. We're going to include horseshoe crabs as well. We'll treat mites and ticks as separate orders. We'll add in amblypygy, and here is that Conychokernes transcriptome in boldface. As we start to zoom out, this green bar is showing you the Conychokernes dataset. Recall that these other 2 pseudoscorpion datasets, which are data poor, are generated from adult tissues. Conychokernes, the developmental transcriptome, has much better recovery of these developmental patterning genes. This is why developmental transcriptomes matter, both as independent datasets as well as resources for annotating new genomes. These white arrowheads are showing you all of the cases where a pseudoscorpion gene is duplicated in common with an arachnopulmonate homolog. And that is a shared duplication. As before, here's the horseshoe crab, independent twofold whole genome duplication, and sea spiders, again, exhibiting no signature of whole genome duplication. Keep zooming out, and you'll notice that across all of the genes that we surveyed, something like 40% of the homeobox genes exhibit shared duplication between pseudoscorpions and the arachnopulmonates. Our third line of evidence comes from microRNA families. And even though the Cryptolechernes draft genome is fairly fragmented, we were nevertheless able to find cases of shared microRNA enrichment that are common across arachnopulmonates and pseudoscorpions. And these are indicated by the pink text. What's more, we found 2 microRNA families that are duplicated only in pseudoscorpions and in scorpions to the exclusion of the other apulmonate arachnid taxa, things like mites and ticks. Our fourth and last line of evidence for a shared genome duplication in pseudoscorpions comes from an extension of Eric Nolan's work. Recall those leg patterning genes like dachshund, where we know that they have these benchmarked arachnopulmonate specific expression patterns. As you might expect, for those 4 genes that Eric surveyed, we found duplicates of all of them. And when we examine their gene trees, we get the gene trees exactly as you would expect after we add in all these new taxa. So pycnogonids have unduplicated dachshund, like harvestmen or akari. And the pseudoscorpion sequences that we find fall exactly within those arachnopulmonate specific clusters. We have one sequence here in red for Doxin 2 for Conicokernes, and we have two partial fragments, two non-overlapping regions for Doxin 1. We're currently using cloning and sequencing to be able to close that gap and reconstruct the entire transcript. It's not just Doxin, by the way. There are many other gene trees that exhibit this kind of signature. And it's very hard to explain these kinds of gene trees and this, more broadly, this body of evidence, if you don't invoke a shared genome duplication that unites pseudoscorpions and arachnopulmonates. So going back to these two topologies, we were evaluating which of these is more likely to be accurate. The weight of the evidence shows that if we compare Hox gene complements, systemic paralogy of gene families, enrichment patterns of microRNA families, or the topologies of genes with known expression patterns and arachnopulmonate-specific duplications, the weight of these independent evidentiary bodies all consistently support the tree on the right. The better sample topology is supported as being accurate. This new understanding precipitates a change in the definition of Arachnopulmonata. We now redefine arachnopulmonates to include pseudoscorpions. The prominent synapomorphy for the group, of course, is that whole genome duplication. This would make pseudoscorpions the only arachnopulmonate order that has completely lost its book lungs. This implies that the tracheal tubules of pseudoscorpions are independently derived from those of groups like derived spiders, as well as other apulmonate arachnid groups like harvestmen, mites, or ticks. Now, you'll notice that the one data class that has failed to recover the monophyly of this composition of arachnopulmonates is, of course, morphology. But without too much difficulty, look broadly across the literature and you will find no shortage of characters that are consistent with this relationship. One example is this courtship behavior, the promenade à deux, which under accelerated transformation under parsimony is reconstructed as homologous across the arachnopulmonates. It's found in pseudoscorpions, scorpions, and pedipalpi. The stock spermatophore, that is the result of this courtship dance, was observed to be likely homologous in specifically attempted pseudoscorpions and scorpions by Oscar Franke in 1979. 41 years later, here we are with an armada of evidence supporting that inference. That stock spermatophore is probably homologous across these new arachnopulmonates. Both that stock spermatophore and the brood sac that pseudoscorpions will produce from gonophoral glands when they lay eggs, these were scored by Jeffrey Schultz as being probably convergently evolved in pedipalpi. But with retrospect, of course, given the weight of all this new evidence, we recognize that those are probably not convergences. We're most likely looking at homologous entities. And we can keep going. Sometimes the most obvious putative synapomorphies are the ones that have been staring us in the face all along. But if there's anything that you've taken away from this talk, I hope it is this: it should not surprise you that we can find morphological characters and realign morphological data sets to be congruent with these other independent data classes. And the reason for this is simple. If there's anything we've learned about morphological characters in deep animal phylogeny over the last 25 years, it is that hindsight is 2020. On behalf of all of my co-authors, thank you for your time.